Hi, everyone. My name is Neil Littman. I'm the founder and CEO of BioVerge, and I am thrilled today to introduce you to Isaac Stoner, who is the CEO of Octagon Therapeutics. Octagon is a really exciting company that BioVerge is looking to partner with. And so I will kick it over to Isaac to go through the presentation, and then we'll save some time at the end for Q&A. So Isaac, I will kick it over to you to run through the presentation.
All right, thanks much, Neil. As mentioned, we are Octagon Therapeutics. And Octagon is focused on developing a new class of targeted immunomodulators for autoimmune disease. So if we look at current treatments for autoimmune disease today, they tend to be non-selective immunosuppressants, things that affect both healthy immune functioning as well as the immune components that are actually causing damage and disease. So Octagon has come up with a novel therapeutic strategy for specifically targeting only those disease-associated immune processes. The Octagon team, as seen here, is based in Kendall Square in the heart of Cambridge, Massachusetts. It's myself and two co-founders, Peter Yu and Fred Ausubel. Fred is a National Academy of Sciences member, spent 35 to 40 odd years at Harvard Med School. Has recently wound down his academic career to help us build this company in his retirement. And our third co-founder, Peter Yu, was actually a postdoc in Fred's lab and is the inventor of our underlying technology. The rest of the team, which includes 3 other PhD level scientists, is focused on biology, immunology, and medicinal chemistry. So if we do look at the problem that Octagon has set out to solve, it's really the poor clinical management of episodic autoimmunity today. If we look at autoimmune disease today, it's characterized by these periods of flare and remission. Disease symptoms kind of come and go, which really complicates clinical management. Typically at induction, patients are placed on a mixture of different medicines, which may include corticosteroids, immunosuppressants like mycophenolate, and then they're slowly tapered off many of these medicines as they can't be used long-term, at which point eventually symptoms tend to flare up again or recur. And this is kind of recurring cycle of trying to get patients on a more optimized therapeutic regimen. What Octagon has managed to do through carefully investigating the basic mechanisms of these diseases is we found specific enzymes that are upregulated in the disease-driving immune cells during disease. And we can leverage these processes to activate our small molecule inhibitors. So this allows us to have medicines that are effectively activated by disease processes, sparing damage to healthy immune processes. And it makes it so that during remission, when these diseases are not active, there's virtually no immunosuppression going on whatsoever, almost a silent drug that's on board only when you need it. So this enables a high level of both temporal and cell type suppression of these disease processes. So if we look at the approach that made this possible, the core observation that was made at Harvard is that immune cells are metabolically distinct during disease. So we spent quite a bit of time in the academic setting and then beyond analyzing exactly what enzymes are being expressed within target immune cells during these autoimmune diseases. We've built a patient sample collection for more than 200 different patients with active autoimmunity, developed a novel screening methodology for teasing out these upregulated processes, and then developed a chemistry strategy to harness these novel biological insights. The chemistry strategy really makes up the core of Octagon's intellectual property position. At its highest level, this is what our chemistry strategy looks like. It's highly modular. The blue group is really representative of the active small molecule inhibitor. We've developed linker chemistry to attach the active to something that looks a lot like an artificial enzyme substrate. It's acted upon by a hydrolytic enzyme that's specifically upregulated in the target cells. So during disease, this enzymatic process will be turned on or upregulated. There'll be a cleavage event that happens on this red group, releasing the active inhibitor within the target cell, at which point it becomes trapped, accumulating within the target immune cells that we know are associated with disease. Octagon's first program is based on a clinically validated target. So that blue group, that active group, inhibits IMPDH, which we know is an essential enzyme for the proliferation of lymphocytes, essential for the proliferation of immune cells. So this builds in a kind of key de-risking feature into Octagon's first program. So in terms of Octagon's patient sample collection that we've assembled, it's made up with more than 200 samples, blood samples from different patients with autoimmune disease. What we've done is we've been able to identify specific mechanisms that are actually conserved across a number of different disease areas. So if we look at our current lead program, we've shown that the biology we're leveraging here, this autoimmune disease associated biology, is associated with 8 or more important autoimmune diseases. So this includes rare diseases such as Pemphigus vulgaris and myasthenia, as well as some really large diseases such as MS, rheumatoid arthritis, and systemic lupus. For a first clinical hypothesis, Octagon has built out a focus on Pemphigus vulgaris. So Pemphigus is a rare disease, an orphan disease, around 150,000 cases per year. It's a severe autoreactive B-cell driven disease where an autoantibody produced within B cells identifies and binds to a specific marker in the skin. This causes severe blistering over a large portion of a patient's skin. This disease has a high mortality rate, still about 5 or 10%, and a lot of this is due to opportunistic infection. So the mortality associated with Pemphigus is actually often due to current treatments that are in use today, things like mycophenolate, rituximab, these non-selective immunosuppressant agents that are being given to these patients in order to get this disease under control. So there is a desperate need in this patient community for something better, something that preserves healthy immune functioning while treating the underlying cause of this severe skin blistering disease. Let's skip up to this. So Octagon's lead compound for autoreactive B-cell driven disease, we call OTG-177. This is the result of a significant medicinal chemistry campaign. And so we now have a small molecule inhibitor in OTG-177 that is highly specific towards B cells only during disease state. So it has virtually no effect on T cells from both a healthy donor or a donor with disease. However, if we look at samples from a patient with active Pemphigus vulgaris, you can see our inhibitor has a very potent inhibitory concentration towards B cells from this patient. IC50 of between about 10 and 20 nanomolar, which really should be sufficiently potent to move this lead program forward. We've also completed a number of pharmacology and safety studies with OTG-177. We've actually dosed as high as 100 milligrams per kilogram in 2 species. And we have shown that this compound is remarkably well tolerated. It's safe in vitro cytotoxicity studies, very stable to plasma and microsome degradation. The compound is currently showing a very favorable PK or pharmacokinetic profile, and we are focused on moving this lead compound forward as a potential development candidate. So if we look at Octagon's current pipeline and development timeline, our lead program is focused on autoreactive B cells, this specific B cell program with an initial application in Pemphigus. So our plan is to continue to optimize this compound through the end of 2020. We have a significant medchem effort going on, supported by a number of CROs. We plan on being in candidate selection in early 2021 rather. And then by mid 2021, we anticipate initiating early IND enabling work. In parallel, Octagon has continued to leverage our discovery methodology. We've run an early screening program looking at specific subsets of T cells, CD8 positive killer T cells, and shown that again, we can identify specific mechanisms that are upregulated in these other immune cell populations during disease. This should be the groundwork for a second program. And then in parallel, we are exploring whether or not there are opportunities to partner with some larger companies around this discovery methodology to form the basis for some joint development efforts. So Octagon's core value creation strategy is going to be focused on this drug discovery approach, our IP filings across a number of different disease areas that cover our novel chemistry. And when we look at value capture, it will be driven through these partnerships. Octagon already has 5 or 6 conversations going on with larger companies. We're continuing to explore whether or not there are opportunities for licensing and for joint development of further programs. The company has been around since about late 2017. Company has closed a seed financing to date, and currently we are in the process of really accelerating a lot of these key medicinal chemistry efforts with an eye towards having that final development candidate in hand and going into formal preclinical development activities sometime next year. All right, Neil, that's about all I got. Thank you very much.
Great, thanks, Isaac. That was a great presentation. It brings up a number of questions as you were going through some of the science, some of the chemistry. There are a number of items I'd like to double click on and learn a little more about. One of them in particular actually relates to the primary patient samples. From our standpoint, we were really excited that Octagon was pursuing primary patient samples and testing the compound in actual patient samples. We think that could be a big differentiator for you and the platform as opposed to just using animal models. Maybe we could just start off, and could you just talk a little bit about the advantages and why you chose to use primary patient samples, and what sort of advantages that could translate into as you think about moving through IND enabling studies and into the clinic?
Sure. No, that's a great question. And so, a lot of that underlying approach came from Fred Alsobel's lab at MGH. And so Fred's lab was deeply interested in metabolism, cell metabolism. And what they found was metabolism is highly context dependent. If you take a cell out of an animal or out of a patient and you put it into artificial growth medium, you'll find immediately that cell will start to have an altered metabolic profile. And all of a sudden won't have to make the same biomolecules that it might otherwise have to make. You'll immediately bias that cell state away from reality. So when we started developing this screening methodology, it was abundantly clear to us that we needed to look at cells, immune cells specifically, in as close to a disease setting as we could. And what's closer to a disease setting than blood, plasma, serum from an actual patient with active autoimmune disease? So that's how we got started down this path of using patient samples as part of our core discovery methodology. We further believe this approach is important as we start to look towards translation towards the clinic. If you look at the animal models of autoimmune disease today, they tend to be highly contrived, right? So these are animals with maybe, for example, something that resembles rheumatoid arthritis, but it's not actually the same molecular mechanisms as rheumatoid arthritis in the human, or something that resembles something like Pemphigus, but really it's a different skin blistering disease. So the ability to show that we can take cells or take blood samples that we've collected from these patients with active autoimmune disease and really have an effect on the target cells within these patient samples, it gives us a much higher level of confidence that our program will translate when we begin looking at early clinical studies.
Great, thanks, Isaac. That makes a lot of sense. Again, an approach that we think is really robust in terms of helping to validate what you're seeing early on in preclinical development, and hopefully that will translate better into the clinic as you move forward. So, okay, great. Maybe we could take a step back. You had a great slide on your team, your advisors. I'm always curious, how did you assemble your team? How did you and Fred connect? You'd love to understand a little more about sort of the history of the founding of the company.
Sure, yeah. So I'm a career entrepreneur. This is actually my fourth go round, and been lucky enough to be in a spot where two of those companies were successfully acquired, had a successful exit. My last company, which was focused on small RNA biomarkers, was acquired in 2010. 2014, at which point I actually went back to pick up an MBA, and the goal of that MBA was to spend some time at MIT in the basement labs trying to meet people like Fred and Peter. So 2 years of focused effort looking at many different technologies. So I actually ended up meeting Peter and Fred through a class that they were involved with at Harvard Business School. They weren't at Harvard Business School, neither was I, but there was a class that's actually very famous in the Cambridge biotech scene now, called Commercializing Science, taught by Vicky Sato, the former president of Vertex, that was offered at Harvard Business School. And the point of that class was to put together a project team around an actual piece of ongoing research. In this case, it was the research that Peter and Fred had going on, with some MBA students, and see if we can build out the business case, see if this is ready for prime time, ready for translation into company formation. So I met them in that class at Harvard Business School in 2015. That was a great low pressure opportunity for us to get to know one another, for me to get to know and understand the technology, and for us to build out a very detailed roadmap for exactly when and how we might take that technology from the academic setting and spin it out into a company. So we took a very deliberate approach. We gave ourselves a few years of continuing to pressure test some of the basic assumptions, continuing to develop the methodology until around the end of 2017, where I was excited enough about the data they had, and so were they, such that I quit my day job at a VC firm and took the plunge with the two of them to start Octagon. So that was about 3 years ago.
Well, that's very exciting, and it's nice to hear the history and that you got to the point where you found the data compelling enough to then start the company, right? So that this is a very data-driven story, which is one of the things that we love about it.
It's part data and then part, knowing that you can get along with your co-founders really well, making sure you trust one another. And so, it does take years to establish that kind of relationship.
It does. That's so true. I think more companies tend to fail by suicide rather than murder, so to speak. So having that founder relationship is critically important. Okay, so you had mentioned you actually took the time. You developed a long-term sort of strategic scientific plan. Maybe we could dive into that a little more. If you think about your long-term scientific vision, let's say over the next 3, 5, 10 years, what are you guys really looking to achieve?
Right. So, we're envisioning this not just as a single asset program. This is a discovery approach, it's a screening approach in chemistry that really does lend itself towards a number of different programs. Our first one being B cell specific, activated by disease processes, that kind of has legs. We know what needs to happen. That's blocking and tackling at this point. But taking that underlying screening approach and beginning to look at, what are the overlooked mechanisms that happen in regulatory T cells or CD4 positive T cells that are involved with a number of these autoimmune diseases? What are the overlooked kind of metabolic hallmarks of disease in these different cell populations that we can use as the basis for program 2, program 3? Really, that's what we're excited about, is how do we go back to the well with this technology, with this approach, and use this to form the basis for a number of really interesting small molecule programs, all based on the same underlying technology. And that's where I think, in terms of disease areas, there are some big, meaty, competitive disease areas that we could begin to chase down with the support of some larger partners that we probably wouldn't be trying to take on ourselves, things like MS and systemic lupus.
Mm-hmm. So if I'm understanding things correctly, it sounds like you're tackling PV as the first indication, orphan indication, limited amount of capital to try to, to really achieve validation for your platform, but it really is an underlying platform that's broadly applicable to PV and a whole variety of other autoimmune diseases, some of which have, very large markets as well.
That is right. And so, PV, that's a way for us to get a win on the board, if you will. I think in terms of building this company going forward as well, we know where our expertise and our passion lies, and that is early stage, right? That's discovery, preclinical, late preclinical, early clinical, and then we're going to be looking to license or partner those programs off. We don't have the stomach for late-term clinical development. We certainly don't have the intention today of building a commercial organization. That's redundant to what a lot of these larger pharmaceutical companies have. Instead, we're going to stay true to really our identity as an R&D organization.
Which makes a lot of sense, right? I mean, focus on what you're good at, and there are plenty of other organizations that have the marketing and commercialization capability. So, you just talked about a little bit the longer term, scientific vision. What you guys are really good at, you're playing to your strengths, right? Is sort of early stage preclinical development. Could you maybe talk a little bit about what that next milestone is that you guys are looking to hit that will help you validate and work towards your overall longer term scientific vision?
Right. So the really near-term milestone for the lead program, so that's the B cell program, is moving into one of these, as mentioned, contrived animal efficacy models. And so we've already done quite a bit of work profiling the immune cells in some lower animal models, some rodent models, to show that the required biology, the required enzymatic processes are present in those rodent immune cells. We plan on going into one of those efficacy models in kind of September timeframe. Currently, we're focused on the PK, understanding the PK and PD relationship of our lead OTG 177 a little better, and continuing to refine a little bit of that chemistry strategy. As soon as we have suitable PK, slow down clearance, have suitable kind of exposure of the target cells to our drug, we're going to be in that efficacy model. And that will be kind of a large step up in de-risking of the program right there. So near term, it's all about animal efficacy model. The next thing will be declaring our development candidate, really making sure we have that drug that we're going to invest in taking all the way through IND enabling work and into clinical development. And so that could, go ahead.
Nope, sorry.
Oh, that actually could be the same compound that we take into this animal model in September. There are just some more boxes we need to check before we declare it as a final development candidate.
Great. And I'm going to put you on the spot here a little bit. Could you talk about what you see as your biggest challenges or your biggest risks to achieving that next inflection point?
Right. And so, for us right now, we're spending quite a bit of time and energy on chemistry, on making sure that our analoging efforts are progressing, and we're kind of slowly optimizing towards this final development candidate. We're doing that on a shoestring budget, and we've been very effective so far. But the risk to us is that program 1 month, or is it 3 or 4 or longer? Making sure that we have the resources to get through that final medchem effort that's probably the biggest near-term risk that I lose sleep over. It's pretty standard to any small molecule program, though, is kind of the error bars on chemistry.
Yeah, makes sense. Okay, so then sort of fast forwarding to assuming everything sort of works out according to plan, give or take a couple months here or there. Assuming you do garner FDA approval, could you talk a little bit about how you're thinking about the target product profile? How your compound would be either used in conjunction with the current standard of care, if you think about it replacing the standard of care? I think that would be helpful for us to wrap our head around.
Right. So I would say, first, we are continuing to refine our clinical hypothesis all the time. We have a clinical advisory board that's getting together in the next couple of weeks to continue to see, are there other orphan indications that we might be overlooking that could be an even better fit for this program. But in terms of how we look at Pemphigus vulgaris and our initial target product profile there, we are focused on developing a small molecule, so orally bioavailable, that is a once daily that patients will use as a maintenance therapy. So typically, the way that the treatment of this disease works is at induction, patients have severe blistering, they're symptomatic, they're going to be put on corticosteroids like prednisone just to get those symptoms under control. And typically that will be effective, but that is a short-term solution. So prednisone can't be taken long-term. It actually has really, really nasty side effects. So we're looking at filling the void of that kind of maintenance therapy that patients transition off of corticosteroids, off of prednisone, onto our oral or our pill that they can take once or twice daily at home. And really, they'll be taking that for life, right? So not only will that be maintaining the health of their immune system, kind of tamping down that aberrant immune functioning, that will also be preventing or prophylactic to future flare-ups or future disease exacerbations. So we're looking at being that convenient, orally available, disease-activated compound that's on board all the time. That's the kind of ideal small molecule maintenance therapy. Mm-hmm. If we look at some of the other competing drugs that are used in this space, these tend to be monoclonal antibodies, things like rituximab. These require really, really lengthy inpatient infusions. So 5 to 7 hours of time in an infusion chair. Those are also non-ideal in that they take many, many months before you see any clinical effect. So when you start a course of these monoclonals, typically you won't see any effect whatsoever for 5 or 6 months. So I think there's really an opportunity to get in there between the short-term solution of prednisone and take these patients and transition onto our drugs as that perfect maintenance therapy that they'll be on for really the rest of their lives.
And so, Isaac, is it a fair assessment to say that your therapy, as envisioned, would be just more highly selective and therefore would have less side effects than some of what's out there today, and so that would lead to, let's say, better patient compliance, less toxicity and things like that. So it's actually a better longer term solution for patients.
So we're actually looking beyond safety. We believe that we should have a much shorter time to clinical resolution as well. So we are actually inhibiting the autoantibody producing cells, plasma cells, that we know are actually responsible for damage, those are not affected by things like rituximab or these other monoclonal therapies, which is why that length to remission is so long. So should be much faster time frame to resolution of symptoms from our drugs, as well as a much better tolerability profile and this maintenance of healthy immune function. So if you look at today, here we are in the COVID era. Always when you hear risk, the list of people who are at highest risk of COVID is patients who are immunocompromised, right? Patients whose immune systems are not functioning correctly. Typically, those are patients with autoimmune disease, and the reason their immune systems are not functioning appropriately is due to the medications they're on. So there's an opportunity for us to be that kind of minimally damaging therapy, where really we're targeting only the disease processes, the immune processes that are involved with disease, and sparing that healthy immune function. We feel that's going to be very, very important in the post-pandemic era that we're in now.
So from the patient perspective, so it really does sound like your therapy is the 1-2 punch of, increased efficacy, plus, better tolerability, a better safety profile. So a much better longer term solution for patients with PV than what currently exists today. Could you also maybe just talk a little bit about, I know you had a slide on, there was a PV specific nonprofit disease foundation. Could you maybe just talk a little bit about sort of the patient perspective and what you're hearing from the needs of patients out there today?
Right. So the group we have engaged with is called the International Pemphigus and Pemphigoid Foundation, IPPF. This is a small group. It is a rare disease, and what we find is, patients with pemphigus and pemphigoid, they may take months or years to A, be appropriately diagnosed, and then B, to transition onto a therapy that really gives them the benefits they're looking for. So it's a long journey for these patients, and the reason that they have this disease foundation is, one, to make sure these patients can get together and have this support network in place, and then to continue to really engage with companies like ours and see what is in clinical development, what are the new treatments that are coming down the pipe that has the potential to change their lives. So we have engaged with that group, and we're going to continue to engage with them and other disease foundations to make sure that we really do stay close to the patient as we push these medicines forward towards clinical development.
And that's really great to hear. I mean, we have a sort of a fundamental viewpoint that it's super important to engage with, the patient. So you're sort of working backward to meet the needs of the patient and not just trying to develop these therapeutics in a vacuum and guess what the patients want. Actually go out there and talk to them. And
right. I think clinicians as well. Super important to engage with clinicians, and we've got some really, really top tier derm rheumatology folks. It's a specialty of a specialty, but we have the right folks around the table to help us as we move this thing forward.
That's great. Well, Isaac, thank you so much for the presentation and taking time for a Q&A today. I think we're gonna call that a wrap. And for folks that are listening and want to learn more, please visit www.bioverge.com. And until next time, thanks for your time.
Thanks, Neil.
Thanks, Isaac. Take care.
Bye.